Hello boys, welcome back to another session of video tutorials, and this is meant for class 12th biology students, and this is for 19th November 2020. As all of you know, we are into the revision mode and we are revising chapter number 12, biotechnology and its applications. And in chapter number 11, if you recollect from your memory, you know that biotechnology is making our lives better, and this is based on two principles. One is the genetic engineering, another is the biochemical engineering. And we also discussed various tools that are required for biotechnology in making rDNA. Biotechnology is basically making a rDNA, recombinant DNA. And in that we talked about the tools like enzymes and other things. Also we talked about the processes. You recollect from your memory 7 steps involved in that, and in that finally we purified the product by using bioreactors. So basically whatever we have learnt in chapter number 11, the purified products obtained from the rDNA technology, now where to apply and how to apply, applications of biotechnology. So in this chapter we are basically dealing with the applications. So biotechnology deals with industrial scale production of biopharmaceuticals and biological using genetically modified microbes, fungi, plants and animals. Its application includes therapeutics, that is various drugs that we use, diagnostics, genetically modified crops, GMO crops for agriculture, processed food, bioremediation, waste treatment. This is the need of the hour, because in cities you have seen, in many of your own towns you are seeing that all the drainage is left untreated and left onto the main roads. This is the sad state of affairs in our country. And then energy production. Whichever country has got high energy production, that is given tag as developed country. And if energy is there, everything is there. Industries will come, GDP will automatically go up. Hence, energy production is a must. The main 3 critical research areas of biotechnology, is providing the best catalyst in the form of improved organism, usually a microbe or a pure enzyme obtained from that microbe. Second, creating optimal conditions through engineering for a catalyst to act. You recollect from your memory, chapter number 9, class 11th, biomolecules. Under that biomolecules we discussed enzymes, and if you recollect from your memory, whenever in a reaction there is an enzyme, it acts as a catalyst by making the rate of reaction faster, and the time in which the reactants or substrates get converted and form products will be minimum. That is the role of an enzyme, being a catalyst. And then finally, downstream processing, which we discussed in our previous session, of using bioreactors, 2 types of bioreactors, so that we get a purified form of protein or the product in the form of organic compounds. So these are the main 3 critical research areas of biotechnology. And biotechnological applications in agriculture. This is because our country is basically an agriculture oriented country. Hence, we need to do lot of research in agriculture. So food production can be increased by, because this is the need, the number of mouths that are to be fed is gradually increasing, and the land that is available for agriculture is drastically getting reduced. So we have got a double-fold challenges, twin challenges need to be met this, and the only solution is biotechnology. So first one, agrochemical based agriculture. Right now we are using lot of chemicals in the form of fertilizers, pesticides, insecticides, fungicides. All these chemicals are entering through food chain into the vegetables and the various products of agriculture, like vegetables and fruits. Basically, when we consume those vegetables, cereals and fruits, the chemicals are automatically, toxic chemicals are entering into our small intestine and stomach. The moment they enter into small intestine and stomach, in gradually, in a phased manner, it's resulting in the formation of the colic cancer, then duodenal cancer, all this. So that's how they are very, very toxic and very, very dangerous. So there is a need for agro-based biochemical forms. And second one, organic culture. Of late you are seeing many people will talk, it has become a fashionable word, organic farming, organic agriculture. But actually we need to give natural things to the agriculture as nutrients, then only we can do something better in agriculture. So our focus will be on that. And the third one, genetically engineered. Basically what is engineering? It is construction. So you construct a new one or damage a old one and construct a new one. So here also what the genetic engineer will do is, he will make a new gene, construct a new gene which is very useful to the crops as well as to the human beings. Or if there is any bad gene, lethal gene we say, dangerous gene, if a lethal gene is there, convert that lethal gene into a useful gene. So this is what genetic engineering will do in agriculture. And thanks to Professor Swaminathan, Dr. Swaminathan, for his forthinking, forethoughts. Green Revolution 1 and 2 were grand success, and they increased the food production many folds. And the basic idea of introducing Green Revolution is better management practices, use of agrochemicals, fertilizers and pesticides. Further increase in production is not possible by using these methods. To overcome this, genetically modified crops are being introduced. So plants, bacteria, fungus and animals whose genes have been altered by manipulation, that's what I said, engineering, manipulation, are called GMOs, genetically modified organisms. Genetically modified plants, GM plants, have many applications. Made crops more tolerant to abiotic stresses. What is abiotic stress? Not biotic. Means it is not related to peer groups. It is related to environmental stress. Like there may be severe heat, there may be loss of water, there may be loss of rains, less moisture or more moisture resulting in fungal attack. So all the salinity of the soil, structure of the soil, composition of the soil, all this put lot of stress on the crop. So we need to make the crops tolerant to this. Second, reduced reliance on chemical pesticides. Just now we were talking about that, the cancers that are formed. And help to reduce post-harvest losses. You all have seen in your own native places, in your own home, that once the harvested crop is stored in your house or in the field, so there may be untimely rains, there may be floods, there may be cyclones, there is no proper conditions to store the harvested crop, rats attack and fungus attack. So lot many factors are there that will drastically reduce the production of post-harvested crops. So how to deal with that? Increased efficiency of mineral usage by plants. See, we were talking about two types of minerals, macro and micro, and even though they are required in milligrams, micro minerals like molybdenum, but they play a crucial role in avoiding many diseases. Hence, their usage is a compulsory one. Enhanced nutritional value of food. See, vitamin A enriched rice, we call it gold rice. It's not enough if we produce rice. Of course, basically all of us know that rice is a carbohydrate, it's a starch. But when this rice is incorporated with vitamin A, it becomes a enriched rice. Vitamin A getting enriched, incorporated into rice will avoid night blindness, as all of us know. So many deficiency diseases can be cured just by eating rice or just by eating any cereal or any vegetable or fruit. That is the enhancement nutritional value of the food through biotechnology. Applications of biotechnology in production of pest-resistant plants. What is pest? It's an insect. It's an organism that eats up the plants. It may eat root so that the entire plant gets killed. It may eat leaves like in tobacco, so the value of the plant gets reduced. And you must have also seen in your own fields a pest eating the stem and putting holes in we call it stem borer. So our area of focus is to make the plants resistive to such pests. So pest resistant plants decrease the amount of pesticides. Bt toxin is produced by a bacterium called Bacillus thuringiensis. Take care of the spelling, thuringiensis. Bt toxin gene has been cloned from the bacteria and been expressed in plants to provide resistance to insects without the need for insecticide. This is the greatness of this. In effect, created a biopesticide. Example: Bt cotton, Bt corn, Bt rice, Bt tomato, Bt potato, Bt soybean. And what is this Bt cotton? Bacterium Bacillus thuringiensis. Bt refers to B for Bacillus, T for thuringiensis. Produce proteins that kill certain insects like Lepidopterans. This is a CBSE question, so you must note down this. Lepidopterans, Colopterans, that is beetles, and Dipterans, flies and mosquitoes. So they get naturally killed. How is that possible? Bacillus thuringiensis produce crystals that contain a toxic insecticidal protein. This toxic protein present in bacterium as inactive protoxin. Remember, inactive. But as soon as insect ingest this inactive form, due to alkaline pH of gut, again condition is there. What is the condition? Alkaline pH. It gets converted into an active form. So inactive protoxin has become active toxin. And once it becomes active, what happens? It binds to the surface of the midgut epithelial cells of the insect and create pores in the midgut, that is intestine, and causes swelling, cell swelling, edema. And once the edema takes place, the cell lysis takes place. The cells will be blasted, and eventually midgut gets blasted, insect gets dead. So this is the great achievement in this. The gene from Bacillus thuringiensis been incorporated into several crop plants such as cotton, maize, rice, etc. So it's a sort of bioinsecticide. The toxin is coded by a gene named Cry gene, CRY. The protein coded by the genes Cry1AB, Cry2AB control cotton bollworms. Cry1AB controls corn borer. Again, you need to remember this. So these are the various achievements of biotechnology. We will continue in your next session, the interesting facts. Until